Welcome to Click for IB Biology. We are studying D3.3 homeostasis. We're moving on to the higher level work of looking at excretion and osmoregulation in the kidney. There's a PDF of this presentation that's linked in down in the description. Download that, annotate this with your own notes, and then why not take the 10-question quiz. There's answers there as well for you to check your understanding of this topic. So this particular video is a general introduction, an overview of kidney function. So of course we have two kidneys, both supplied by the renal artery. We have two renal arteries and we have two renal veins supplying each of the kidneys on either side of our body. The kidney has two major functions, that one being excretion and the other one being osmoregulation. Excretion we can think of as the removal of metabolic metabolic waste molecules. So for instance, we could have things like carbon dioxide, but of course carbon dioxide is largely excreted from the lungs. So for the kidney, the major molecules that need to be removed are urea, which is the nitrogen component of amino acids, or indeed another version of this is uric acid. So these two compounds here are essentially the removal of the nitrogen component, as we cannot actually store things like amino acids. The second function of the kidney that we'll be taking a look at, and in fact most of our attention will be paid to osmoregulation, which is the regulation of the osmotic concentration of the blood plasma and indeed therefore of tissue fluid. Now what this actually means is if we're regulating the osmotic concentration, so we know that osmosis is going to be about regulating water content, but of course there's two ways to alter the concentration of a solution, so we can also regulate solute levels as well. So through the regulation of water content and solute content, we can regulate the osmotic concentration of the plasma and tissue so that it will remain isotonic with our tissues, with our cells. So let's take a look at this. As we go through this part of the video, I'm going to be relating the structure of the kidney in a descriptive manner to its function, and then in subsequent videos we will look at the explanatory part of this process. So within a kidney, within a human kidney, there's anything between 1 and 1.5 million tubes called nephrons. And the distribution of the sections of this nephron account for the coloration that we can see in the cross-section of this particular kidney that you can see here. So we have this outer caramel layer, which is the so-called cortex. We have the medulla or medulla region there, which forms almost pyramidal features, so pyramidal medulla. And then there's this white area here is where the urine actually collects and drains in ureters to our bladder. Now, if we relate that to the actual nephron structure, each one of these nephrons has, let's look at its input of blood supply is called the afferent blood vessel. The blood leaving is known as the efferent. The nephron could better be thought of as a dead-end tube. So let's actually approach it from here. This is the opening of the nephron, and if we locate that on our diagram here, that would be on the edge of the pyramid here, like so. And coming out of this tube here, which is called the collecting tubule, we would actually have urine. And so the kidney's done its job, and the urine therefore would emerge from here, here and here and here around these reddened edges and collect in this space here and drain away as previously thought. Now, if we follow that tube upwards, we're going through the collecting tubule, which is here, and that comes up and into the cortex, where it begins to twist and turn. And this first twisting and turning is called the distal convoluted tubule. Then what happens is it dips down into the medulla again and then back up, and this dip is called the loop of Henle. Once the loop of Henle comes back up into the cortex, it then does a further twisting action, which is known as the proximal convoluted tubule, terminating in a dead end known as the Bowman's capsule, which is this structure B here in the diagram. So that's the Bowman's capsule. So of course, interestingly, if we look at the Bowman's capsule, instead of ending in a dead end like so, it actually becomes enlarged and then pressed inward to form a cup-like structure, difficult to draw in 2D, like so. And of course, that's partly to increase the surface area necessary for the filtration process. So we're going to look at the effects of each of these different tubular sections. Each has its own function. So on the right hand side we have the nephron again, and you'll notice that the letters are clearly shown here, A, B, C, D, and these correspond to my flow diagram on the left hand side, which will summarize the filtration process, which actually brings about the removal of toxic waste and excretion, but also the regulation, the osmotic regulation. So we begin with blood, unfiltered blood coming into the kidney in the renal artery. The renal artery branches, of course, down into arterioles, and the input going in here, A, is the afferent, let's draw that, afferent blood vessel, and that enters the Bowman's capsule. And so we have the afferent hit. So A here corresponds to A here on this diagram. Within the Bowman's capsule, the afferent blood vessel begins to form a tightly knotted blood vessel. This structure here, I'll just label this individually, is known as the glomerulus. And it's the combination of the glomerulus and the Bowman's capsule that creates our filtration unit and a process which is known as ultrafiltration, which is at section B, which is here. So the filtration process, interestingly, what happens is that the filtrate, which is now forming in the nephron, so having passed through the filter of the Bowman's capsule, blood is filtered, and the filtrate, which is formed, is called nephron filtrate. And this is going to pass along the length of the entire tubule. Initially, at D, the filtrate is non-selective. So ultrafiltration is not a selective process particularly. It allows through most molecules which are soluble, including things like glucose, water, amino acids, uric acid, and urea, but no proteins are able to enter this stage, and no cells get through at this stage. The filtrate ultimately will emerge from point F, which is going to be our urine. But before that happens, the efferent blood vessel here winds itself around the nephron in such a manner as it lies in very, very close proximity. And what's going to happen, if I could just summarize essentially, is that if the nephron is here and the blood vessel is here, this is the efferent blood vessel, I'll put E for here, and this is the nephron here, molecules are going to be selected and taken back into the bloodstream. So having had a non-selective ultrafiltration, what then happens is that we selectively reabsorb molecules back into the blood, and in this way we bring about the regulation of blood. That regulated blood will then pass back into the renal artery. Anything which remains in the nephron forms urine. And so our urine will contain combinations of water, urea, and uric acid. So let's look in other videos at the detail of each of those stages in the process. And so the link to ultrafiltration is appearing on the left, a chance to subscribe to the channel in the center, and the playlist is on the right hand side.